Okay, so as Olga mentioned before, we are QTSense. So in QTSense, we developed a device that we call Quantum Nova. So today I'm going to introduce you how this equipment works and also what we do in our company. And I titled this presentation the Quantum Biomicroscopy for Life Science Applications. So, who we are? So, we are a precision engineering company located in the Netherlands, specifically in the city of Groningen, in the north of the Netherlands. And we are specialized in the design, development, and deployment of the quantum-enabled microscope. So, basically, the Quantum Nova platform that we commercialize in our company. Why do we matter? Basically, because we reveal the earliest biological response related with free radicals kinetics, enabling faster drug development and new scientific discovery and better diagnostics that with the purpose to save lives. And for us, our vision is to lead the world in quantum biosensing and redefine how biology is measured. And our mission for in our company is to reveal the earliest biological response and turn them into the real world impact for research, medicine, and society. So I'm going to show you later on in my presentation different specific applications that we are working with in this moment. And I want to highlight one of them that in which we test a specific anti-cancer drug, and we were able to detect in the early beginning of the process how this drug is working, basically correlating the effect with the free radical generation. So this is the Quantum Nova. So basically, our device is a high spatial and temporal resolution microscope. It's a modified confocal microscope that is specialized in the measuring of free radicals generation neutralization. So basically, with our Quantum Nova, we can measure the emission using fluorescent nanodiamonds that are provided by you at Amos Nano. Basically, this is the biosensor that we use in our technology, and we can do this kind of measurements in real time. We can do that in the high temporal resolution. It means that we can do the free radical detection in a continuous monitoring of those free radicals in longer period of times or in shorter period of times, depends of the design of the experiment or depends of course of the what is the requirement of the customer of what they want to do related with the experimental design. Also, we do that in the highest spatial resolution. So basically, we can measure the free radicals around the nanoparticle in 300 nanometers, and we do it in high sensitivity. It means that we can measure concentrations of those free radicals around 1 to 10 nanomolar. These sensitivity or these levels of detection are correlated with the physiological production of those free radicals. So it means that we can do that in also in high sensitivity. So what are the free radicals? So basically the free radicals are molecules that are commonly produced in the cells, inside the cells or inside the biological samples. And one of the main characteristics of these free radicals are that in the latest spin of the atomic structure, they have a missing electron. So this missing electron can interact with the lattice structure inside the nanodiamonds that we use as a sensor in our method. But why they are important? So basically, the free radicals play a key role in diseases development in the human body because the three main functions of the free radicals inside the cells are related with the cellular signaling, the defense against the pathogens, and also the homeostasis process. So usually the free radicals are related with the maintenance of the homeostasis. It means that when the cells produce the free radicals, basically they can regulate different biochemical processes inside the cell and are commonly produced by the cells itself. So what it means that when any of these levels of free radicals that are produced inside the cells exceeds the body ability or the cell's ability to neutralize them, we can call this kind of a state of oxidative stress. It means that we're going to have a lot of free radical production that create an imbalance inside the cells and leads the process to generate different diseases. Which kind why do we care about the free radicals? Because this imbalance or this change in the free radical concentration that I mentioned before could change could affect the DNA, also the protein oxidation or even the lipid oxidation. So if you see, the free radicals are related with the main biomolecules that are inside the cells. So it means that all of this process gonna lead the different kind or the different main diseases that are well known. For instance, the cardiovascular disorders, autoimmune and neuronal conditions, cancer, diabetes, and obesity that are related usually with the, for instance, the endothelial cell damage in the cardiovascular disorders, chronic inflammation with the autoimmune with the autoimmune disorders, DNA mutations in cancer, also the microenvironment that is created inside the tumors, hypoxia, inflammation, and as I mentioned before, the lipid oxidation or lipid peroxidation, that is one of the main processes that is related with the diabetes and obesity development, and also in liver, correlated with liver diseases. Um, there are different challenges for the free radical detection. The first one is the short lifespan. So it means that the free radicals usually are highly reactive, and usually they last only, they are produced and consumed just in microseconds or milliseconds, making the real-time measurements difficult. So also the low concentration of those radicals inside the complexity of the cell interferes sometimes with the standard detections. Uh, Beside the complexity inside the intracellular environment, those free radicals usually are present in very low concentrations, usually in the physiological level or of nanomolars, and they require high sensitivity detection methods. The lack of selectivity with the current standard methods that are used for the detection of free radicals, or with reactive oxygen species that are usually associated with the oxidative stress, also becomes a really high challenge to try to understand what is the real production or the specificity to distinguish between free radicals species. Usually the methods that are commonly used to detect those free radicals are based in the labels, or detecting the damage, or usually detecting the cellular response. So as you see here, usually we cannot detect with the standard methods direct responses associated with the free radicals or the oxidative stress. So that's why we have plenty of these methods to do the detection, even for instance, in specific locations like mitochondria, using mitosocs, are also using different technologies like flow cytometry or spectrophotometry or even HPLC to detect, for instance, different responses or different components that could be associated with the free radical production. So there are different advantages or and disadvantages of these methods. For instance, for the fluorescence proofs, the high advantage is that they are really cell permeable. The intensity is quantifiable, and also the product usually is stable. But one of the main disadvantages of that fluorescent proofs are the complexity of the products that usually interferes with the specificity of the detection. So it means that also these interactions with 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 the products that are related inside the cell could affect the measurement itself. So in general, there are not conventional methods that are available to simultaneously real-time measure and localize and quantify the free radicals, even at subcellular level with high sensitivity. So that's why in our company, and also based on the research from Romano Shirgahol group, we use quantum sensors to detect the free radicals in real time at high spatial and temporal resolution via the magnetic moment of those free radicals. So basically that we use is this free electron that I mentioned before at the beginning of the presentation to correlate and detect this free electron as a magnetic noise inside the cellular environment, and do an interaction with the nitrogen vacancy centers that we can find inside our nanosensor. So we use the nanodiamonds as the quantum sensor. So basically, we use the impurities inside the diamond lattice that you will know, and maybe that you are expert also in the understanding how these MB centers works. But basically, here we use the structure, the MB centers that are replaced by a carbon inside the nanodiamond structure, and these electrons in the vacancy are sensitive to the magnetic moment. It means they are sensitive to the free electron that we can find in the free radicals that are produced inside the biological environment. So we use these nitrogen vacancy centers in the diamond to convert the magnetic resonance signal. It means the magnetic resonance noise that are produced for the free radicals into light. So we transform this magnetic noise into a fluorescence proof using the own characteristic of fluorescence of these nanodiamonds. And we use this to measure the free radicals in living cells without affecting the natural state of the cell itself. So it means that these nanodiamonds are bioinert, and also they are not cytotoxic. We have plenty of papers already published where we test and where we know that those nanodiamonds are not affecting the viability of the cells, or that not interferes with the biochemical process that we want to study as well. So that's why we use these defects inside the nanodiamonds to correlate and measure the amount of free radicals that are inside the biological environment. So basically that we use is the fluorescent, in our microscope, we have a green laser. With this green laser, we excite the nitrogen vacancy centers that are inside the structure of the nanodiamonds, and we get an emission as response from the MB centers. So it means that when we excite or when we are polarizing the electrons in the of the MB centers, we can get an emission and measure the time, the relaxation time that takes for this electron to return to the ground state, and correlate that speed of relaxation with the amount of free radicals that we can find inside the biological environment. So usually we measure in our device different dark times with 10,000. Usually the standard method that we use is 50 dark times and 10,000 repetitions per dark time. So that we do here, as you see in this figure, is we polarize first with the green laser the MB centers. Then during the dark time, we measure the first 5 microseconds after the polarization. And then we do subsequent emission process readouts during each dark time. So then at the end that we're going to have is a relaxation curve that will give us an approximation, or will give us a T1 time that we can correlate with the free radical intensity and with the free radical concentrations inside that environment. So the free radicals, as I said, affect the intensity and dark time correlation and lower the T1 relaxation time. It means that when we have lower free radical amount inside the biological environment, we're going to have a longer T1 time because gonna takes longer time to do the interaction, and we're gonna have a T1 result longer uh, in this measurement. So on the other way around, when we have more free radicals inside the cellular environment, we're going to have shorter T1 times because the speed that takes the electron to return to the ground state going to be faster. So in this way is how we can correlate the free radical concentration with the generated inside the cellular environment using the fluorescent nanodiamonds. So how we do it? Basically, we introduce the nanodiamond inside the cell, coated or uncoated. What it means that also we can functionalize the surface of the nanodiamond with different biomolecules. Here in this figure, you can see an example that is commonly used in our lab, where we coat the nanodiamond with BDAC2, that is one antibody that direct the nanodiamond to the outer membrane in the mitochondria. The mitochondria is one of the main producers of free radicals inside the cells. So using this strategy, we can identify the free radical generation nearby the mitochondria and, for instance, measure a different mechanism of actions of small molecules to control different kind of cells, or also we can measure the antioxidant effect or different reagents as well. And for cells, we commonly use 70 nanometer nanodiamonds in size. And for larger samples or larger or complex samples, usually we use 120 nanometers, for instance, for tissues. So we use the own mechanism of the cell to internalize the nanodiamond. It means that the cells has different mechanisms to do this kind of internalization. We use the endocytosis process that the cell has to internalize the particle. It means that we're gonna use different times and also different concentrations of diamonds dependent of the type of cell that we're gonna use. For example, if we are working with macrophages that are one of the main immune cells that we have in the system, usually these macrophages does the internalization usually in 1 to 2 hours. But if we are using liver cells, HepG2 cell lines, or adipose cells, for instance, we have to do the incubation overnight or even 24 hours longer. So it means that we gonna use the own mechanism of the cell to do the internalization, as I said before. And after that process, we can create, for instance, the induction of different process. We can create an stress with any inducer or uh, add any kind of treatment. And then at the end that we're gonna do is compare the baseline process that we measure in the cell without the treatment and after the treatment, or after the induction or after the inhibition. So it depends a lot what is the experimental design that is related with the application that we're gonna do. Then in this process, so that we are doing is seed our cells in the Petri dish. Usually we use Petri dishes with glass bottom Petri dishes to help the cells to be to start the adhesion process in the bottom of the plate. Then, after the incubation after the cells are attached in the bottom of that Petri dish, we add the specific concentration of nanodiamonds, and we put the cells under incubation again during the specific time that is allowed to this specific cell line. and basically to provide the cell the environment to do the internalization of the nanodiamonds. Then we place the cells with Petri dish inside the quantum nova. Oh, sorry, my presentation. Yeah, inside the quantum nova, and then we start with the standard settings that usually we use to do the measurement, 50 dart times, 10,000 repetitions, and then we're going to get the output, as I mentioned before. So usually we measure the baseline, and afterwards we measure the result after the treatment. So it means that after that, we can measure the low concentration or high concentration of those free radicals generated inside the cell and associate that process with different diseases or with the effect that we are looking for dependent of the treatment that we are adding to our cells. So in summary, that we are doing is use the nanodiamonds with MV centers inside the cell environment. We use the green laser that is inside our confocal microscope and use the MV center. The MV center is gets excited and starts to spin and interact with the magnetic noise of the free radicals inside the inside the cellular environment. Then we quantify the quantum phenomena using the tiny quantum sensors that detects and measuring the free radicals based on the electron relaxation time. What is the advantages? What are the advantages that we have here? So basically, we can talk about the sensitivity. Using the quantum sensing, the sensitivity is really high because we can detect the free radicals in real time at subcellular resolution, as I mentioned before. Not only in mitochondria, that one was one of the examples that I mentioned, but also we can do it in nuclei, endoplasmic reticulum, or different organelles inside the cells. If we compare our method with the fluorescent ROS assays, usually the sensitivity in those assays are moderate. The sample volume is really low in our case compared with the others, and also the interference is low. The measurement type, we can do that in real time and continuously. That help us basically to detect the free radical kinetic process inside the cell and check what is the real effect of the treatment that we are adding to our inside our cells, and also define, for instance, early detection of different diseases. One of the projects that we are working right now is detect, for instance, the free radicals associated with sepsis process in the intensive care units in the ED department here in the UMCG. And we are able to understand and differentiate between healthy patients and patients that are or that are starting or developing the sepsis process based on the free radical detection in the early stage of that process. Also, our method is compatible with tissues, and the reaction dependency is really low because we don't, with the nanodiamond or with the laser that we are using to excite the MV centers inside the nanodiamonds, we are not generated any kind of stress in the surrounding area. It means that we can detect the real production of free radicals inside that kind of cells. So let me move forward to different applications that we have already working. So the first one is related with cells. So here is a paper that was published in 2024, where was a study the stress induced by hypoxia in single heart muscle cells. So in this specific research work, we coated the nanodiamonds with the antibody BDACT to direct the nanodiamond and check the free radical generation directly in mitochondria. So then, after this process, we put the cells under hypoxia and we measure it before and after the hypoxia process, what happened with the free radicals generation. So if you see here in the figure A, this is one of the standard methods that didn't show us a big difference after the hypoxia process, even at 24 hours. But if we use our technology based on nanodiamonds, we can see the difference after the 24 hours, and we can see the increasing of free radicals associated with the hypoxia process. Then, if we observe the impact of the hypoxia inside the cells, we also treat those cells in a reoxygenation process, and then we follow and we track the free radical generated in different times, each 20 minutes during 3 hours. We was measuring what happened with the free radicals when we introduce again oxygen inside the cells. And as you see here, we can see a big difference, and we were able to track the free radical generation during this time and correlate the after 3 hours. Basically we can define that the cells were recovered in the healthy process compared with the controls, and we were able to establish the homeostasis of the cells, and we were able to see how the homeostasis in the cells were relocated based on the free radical generation, based on the free radical tracking associated with this kind of cells or with this kind of process. Also, we can do that in fluids. So in this, for this kind of application, the method is quite different. Usually that we do is dried before, and a specific concentration of the diamonds usually is 5 to 10 micrograms per ml in the surface of the Petri dish. And after that, we do some measurements in the dry state. And in this specific case, we added synovial fluid. So when we added the synovial fluid, we were able to measure the free radicals associated with the synovial fluid of patients with arthritis, and test one drug that is called piroxicam, that usually is commonly used to reduce the effects of the arthritis inside the patients. So here with this method, with this process, we were able to differentiate between osteoarthritis patients and rheumatoid arthritis patients, and check what happened after, if we compare the dry diamonds uh, before the addition of the synovial fluid. You see how the synovial fluid is showing a high concentration of free radicals. And after the addition in this same sample, we see how the free radicals goes up, and then help us basically to control the free radicals associated with the arthritis process in the synovial fluid. So in general, with this study, we were able to identify that using our technology, we can test how the drugs associated with these specific diseases can control the free radicals itself. And if you see, for instance, in the rheumatoid arthritis patients, we didn't see the same behavior. So it means that the piroxicam is much better to treat patients with osteoarthritis instead the rheumatoid arthritis. So this is the level of translational process that we can conclude with our device and with our technology, and also basically help the clinicians to understand how these kind of drugs can work, and also how they can prevent the disease or work directly with different patients. Another kind of samples that we can use are tissues. So in this paper, basically, we was testing in precision cut liver slices. We took in this specific project, we took the liver from from mouse. And then we use a technique that is called precision slices, where we were, and using this technology, we were able to cut in small portions of this slide and culture the small piece of the tissue in a petri dish during 7 days. Then, after that process, we remain alive the tissue under these conditions, and we were able to test different two components. The ethanol that usually is associated with the damage of liver in humans. And also then we tested one antioxidant. In this case, we did it with ascorbic acid. So if you see in the figure in gray, is the in this process we did the tracking of the free radicals generated in the tissue without any kind of treatment. So that's why we can see the behavior during the time after 33 minutes. And then we added the ethanol. So when we added the ethanol, you can see how after the addition of the ethanol, the curve drops. It means that the free radicals were increased in the process. And if we add the L-ascorbic acid to the same sample, you can see how the curve is increasing. So it means that we can control, we can observe not just the effect of one stressor in the sample, but also how one antioxidant, in this case of one drug that can help us to control the process, gonna help and gonna increase also the free radicals. And we can associate all of this process with, in this case, with the liver diseases. Finally, this is one of the recently process that we did in QTSense, testing the mechanism of action of one drug to control breast cancer cell. So breast cancer, sorry. So here that we used it was breast cancer cells, MDA MB231 cells, and in this one, we track also the free radicals before the addition of the anti-cancer drug. And as you see here in black is basically the control before the addition of the drug, and in red after the addition of the drug. So if you see, as soon as we add the drug to our cells, we observe how the free radicals increase this process. What it means? It means that we can correlate the effect of this drug in the early process that leads usually the metabolic process to the apoptosis. So with this drug, we certainly define or verify in the early detection, just in 3 hours, how the free radicals are increasing the damage inside the cell and leads to apoptosis. And usually we associate also the mechanism of action of this drug that we was using here is related basically with the inactivation of one specific protein that that does this process, that after the blocking of the protein, basically the free radicals are generated and then leads the apoptosis process to kill the cells. So it means that here we can detect in the early process how the drug is working, because these researchers were using the same small molecule to check inside the cancer cells, they detect the effect of that drug just after 24 hours. So it means that here we can also use our technology to understand how the drugs are working in the early detection and provide, for instance, pharma companies a quality control process for the drugs, and basically check in the faster how those drugs are working in the specific mechanism of action that they are looking for. So just to move forward and go to the final part of my presentation, I just want to show you a little bit what is the design, the optical design of our instrument. So basically this is the internal design of the Quantum Nova. So basically we have a motorized 3D stage. All all of our instrument is completely automated. We have the filters and pinhole detectors, laser source, the lenses that help us to filter in the light also that and also usually if we have high background samples, we can use these lenses and these filters, sorry, to decrease the background that came from the sample and separate, for instance, the spectrum that can affect, or the noise that we, and isolate the noise that can come from this sample, and then just detect the intensity that came from the diamond. We have the laser monitoring system, the dichroic mirror, that separate basically the signal from the green laser and the emission that came from the nanodiamond, and the galvo mirror that control the scanner process that help us basically to do the scanning of our sample. So this is the architecture of, in general, of the different components that we have inside our equipment, the sample movement that is completely automated, the scanning, excitation, emission detection, and a camera with a bragg field. Also, for sure, we have a software that help us to do this kind of detection. So we have 10 core functions inside our current software. We're also working to improve the current version that we have. And basically, that we can do is find, we have an automatic FND finder that localize and test and help us to test for photo bleaching inside the cell, and help us to identify the nanodiamond that we have in our sample. We have a complete manual control is possible as well in the 3D stage. Currently in the coming version that we're going to produce, everything will going to be automated. And for sure, we have an automated data analysis and visualization. This is a short video that show you in live how the system works. So basically, we have here the confocal image and the bright field image. We have the scanning process where we can scan the larger area inside the sample. Then we can basically localize after the scanning one specific nanoparticle. Here you can see the particle. Usually we do focusing process to place the nanodiamond in the center of the image, of the confocal image. Then we can measure the real photon count that came from this specific nanoparticle. We check if the particle is stable to do the measurement. Also, we can save the particles for future analysis if you want. So it means that we can, if we move to another 2, 3, or 4 particles, we can go back again to the previous nanoparticle and measure if, let's see, let's say that we are doing measurements of different concentrations. And here is the curves that we provide with the T1 number, R square, and the data analysis. So then after this, we can collect basically the information and export the information in a CSV file and do the comparisons afterwards in another statistical system, for instance. So this is the unique the parts, the items that are placed with our system. Basically, we have the base unit, the consumables. Also, we offer a maintenance contract and additional subscriptions for a license, future license of the software. So for sure, we are open for collaborations. We have demonstrations online, and we, as I mentioned at the beginning, we are going or participating in different events. Many of them here in Europe, but the next week, Deepak gonna be our CEO, gonna be in Boston, also talking about our technology. And we are open for different exploratory projects where we can test our device and create strategic partnerships with different customers. So that's it. And this is my presentation. So thank you very much for listening. And I'm open for your questions now.
Elkin, thank you very much. It's very exciting presentation. And we have first question. Please raise your hand or start asking the question. So let me start with my question. Sorry, there is Saravan. Please ask your question. Saravanan.
Hello, can you hear me?
Yes.
Okay, thank you very much for a very interesting talk. So my question, I have two questions. One is, how do you convert this radical concentration, determine the radical concentration from the T1 measurement? How does it scale, like a linear or kind of what model it follows?
Yeah, this is, yeah. Yeah, go ahead.
Yeah, and the second question is, can you identify the type of radical, for example? Like, you have different types. So
Yeah.
something like oxygen ones or hydroxyl ones.
Yes. So to answer your first question, usually we can correlate the concentration of those free radicals based on usually we use as a standard gadolinium. Gadolinium is one of the strongest paramagnetic species. So usually we create a standard curve using gadolinium, and with this gadolinium we can test what is the sensitivity of our instrument. So then we can correlate. We have also some papers that I can share with you if you are interested in reading that part as well. But we do is basically correlate this standard curve with gadolinium with the free radicals that are produced inside the cells. So this is how we can measure the free radicals, or we can correlate the concentration with the free radicals using this standard curve. And secondly, unfortunately, for us at the moment, it's not possible differentiate between species. So basically, we measure the whole amount of free radicals that are in the cellular environment. And yeah, but one thing that usually we do is use different biochemical process to correlate more ROS species related with the specific with some specific process. Let's say that you are using one knockout cell that help you to understand how the ROS species are produced. So then you can correlate that process indirectly. But in general, it's not possible with the technology do the differentiation between the different radicals that are produced. So we measure in general the whole amount.
Okay, thank you.
Yeah.
Thank you. Next question is from Jonathan. Jonathan Owen, please.
Hi there. I have some questions about the distribution of T1 times that you measure. So you have these nice plots where you look at the spread in T1 and, change the chemical composition and then measure a spread again. And I wondered if that was different nanodiamonds that are giving that spread. And then also, how reliable is the T1 for a given nanodiamond? In other words, if you change the environment of that crystal, does that influence the T1 time without the introduction of radicals? And I guess the real question I'm after is whether the T1 is uniquely sensitive to free radicals or whether it's also sensitive to things like photochemistry of the sample over time.
Yeah, this is a really interesting question because usually okay, let's say that in terms of the other kind of species that are related that can generate or interact with the diamonds, yes, maybe we can measure that because we are measuring the whole amount of radicals or a species with free electrons. So it means that we can measure also different type of species associated with that. Yeah.
Yeah, I guess I'm wondering also if that T1 is uniquely sensitive to the free radicals. So, you add ascorbic acid and ethanol, and then you see a change.
Yeah.
And the claim is that, I can see that the cell is harmed by this or that it's healing, because the assumption is that, your measurement is entirely sensitive to free radical concentration. But I guess I wondered if that's true.
Yeah, I know it's true, because also so in general that we can do is test also use different controls with additional and well-known antioxidants, for instance, and then compare the effect that we are seeing with our testing method. Let's say the ascorbic acid of the ethanol. For instance, when we want to check what is happening inside the mitochondria, usually we use menadione to test as a standard to check how the free radicals are increased in the mitochondria, and then do the comparison with with the different stressor or inducer that you are using. So we have to control it using well-known controls that help us to check that really the free radicals are generated because the stress that is created by these kind of compounds. So usually we do it and we compare it with the controls, with the well-known controls.
There's a paper Peter Maurer published on the zero field splitting during a measurement, and they looked at nanodiamonds with and without a silica shell. And what they find is that the zero field splitting changes with time in a way that is not easy to understand. And it's less sensitive when there's a shell. And their explanation from this is that there's something happening to the diamond with time that's influencing that energy. And I could imagine that changes in T1 would also accompany that. And it's not, I took away from that study that actually this quantity that you're measuring is sensitive to other things that may not simply relate to free radicals and might involve surface chemistry of the particle, for example.
Yeah, indeed. Yeah, thank you. This is a really nice discussion. Yeah, this is a really because also this is something that usually we have to check, because indeed is I know the paper that you did mention, the one with the core shell. And also in another papers is know that when we coat the diamonds, for instance, with polymers or with another kind of these chemical structures, we can the T1 could be modified, but it's also because the intensity of the diamond drops a bit when we do this kind of coating. So yeah, but yes, this is something that we have to test as well.
Elkin, if you measure T1, how large variations would be, say, if you measure the same particle over 1 hour? Will T1 of a particle inside cell or without any stressors, just nothing is changed and you look at T1 only, what are variations?
Yeah, usually the variation with one particle, we have some statistical analysis on that, and it also depends a lot. So we have variations between particles, and also we have variations in repetitions per particle. So usually the threshold, the threshold per particle is usually between 5 till 20. So the variation is that the spectrum is, in my consideration, is a little bit high, but we have, depends a lot of the particle that we are measuring, but we have usually this kind of variation, less than 20%.
And there is also instrumental noise, right? There is drift from instruments, from your, say, laser, maybe AC in the room or something else. So did you look, say, for sources of instrumental noise and if they impact on drift of T1 measurements over time for the same particle?
Yeah, indeed. So this also the influence of the light in the environment could affect also the change. Maybe from the biological samples, also the background from the sample could affect a bit the variation. But we did a lot of tests also per particle, and sometimes also we expect some kind of Brownian movement of this nanodiamond inside the cytoplasm or inside the organic fluid in the cell. So we are expecting some kind of movement. That's why we use the confocal microscope basically to localize and put the coordinate in the specific location. Mm, but yes, the light could influence a little bit the change, but I did plenty of tests with different particles, and this kind of variation is usually the same. So it's less than 20%. In some particles could be better, maybe just 5% in variation, but in other ones could be higher, but not more than 20. So this is this is like the threshold that I measure. Also, when we do comparisons between particles, the variation is also remains in 20. It's not going higher on that. So this is something that we have data to demonstrate that this is a result. Yeah. Yeah,
Do
but
we
we
have
at
more
the end we
questions?
expect some kind of variation in the particles itself. Yeah.
I also have a question related to endosomal escape, right? Since you have this endocytosis mechanism, how endosomes and freeing from endosomes is implemented. Because if you have endocytosis, right, then diamonds are in endosomes. After endocytosis, and then what happens next? How you get proximity of free radicals?
Yeah, usually when we have the endosome, as we that we know based on the confocal standard images is that the nanodiamonds are depends if you are not coating the diamonds, so you expect that the diamonds are randomly distributed inside the cytoplasm of the cell. And then we have also papers published where we know that the nanodiamonds usually escape after 7 to 8 days. So the nanodiamonds remains inside the cell after that time. In some cases, depends of the type of cell that you are working with, as they escape faster, or maybe they can remain longer inside the cell. Yeah, but usually, if we are not coating the nanodiamond, we expect to have it randomly distributed in the cytoplasm and remains for this period of time. But then if we coat the nanodiamond, for instance, with the BDAC, that is the example that I gave you during the presentation, also we can see based on based on the standard confocal images that the nanodiamonds are really located, and because we measure the Mander coefficient, and we do the colocalization analysis basically to check what is the amount of those nanodiamonds that were introduced that are colocalizing with the mitochondria, for instance. So we know that in some cases, in the HepG2 cells, for instance, usually we have a Mander coefficient around 0.9. And in cancer cells, usually is also 0.8. So it's variable. We have some kind of variation between the cell type that we are using. But yeah, but the endosomal escape usually is related with the process that we are using, if we are coating or not the nanodiamond. And yeah, but with the results that we have, usually we know that they remains long time inside the cell. We can detect it even after 7 days.
Okay, great. Do we have any more questions from the audience? Please feel free to just start asking.
Yeah.
What is statistics? How many nanodiamonds you have to measure, say, for some particular study in order to say, oh, it's, say, strong statistics that I make my conclusion?
Yeah, usually that we do is try to measure, depends of the study for sure. Uh, mm, Usually we try to measure at least 10 diamonds per repetition. So it means that at the end, if we do the standard, that is 3 repetitions, 3 biological repetitions, so we're going to measure at least 30 nanodiamonds per treatment or per group or, yeah, per experiment. So I would say that with 30 nanodiamonds, we're going to have a strong statistical comparison that will give us strong conclusions, and yeah, and then see the behavior and what happened before and after the addition of any kind of drug or stressor.
And do you look also at different cells, at few cells?
Yeah, in
And then
different
say for
cells.
how many cells? Is it 30 nanodiamonds
Okay.
per
Yeah.
cell? So kind of say
Yeah,
when you
usually
study some
we try
particular problem.
here we measure in single cells. So we usually that we do is measure 1 diamond per cell. It means that in each repetition we are measuring at least 30 cells.
Okay.
Yeah. Yeah, because also depends what is the size and shape of the cell. We can find more than 1 diamond per cell. So if it's interesting also for the process to check and check the repetition in just one cell, we can measure different nanodiamonds located in just one cell and maybe just decrease the number of cells during the design of the experiment. But usually that we do is measure 1 cell at the time and 1 diamond per cell.
And then say in the case if there are heterogeneous cells and then some diseases or some other applications have some cells subpopulations, then can you kind of identify those subpopulations? And then probably you need higher statistics for that case, right? Maybe 100 cells at least, so that you can see trends.
Yeah, this is a really nice question because currently all the process that we did until now was based just on in one cell culture, cell line at the time. So we didn't test in co-culture or in different cells that are located in the same sample. One strategy that we are trying to test on this process is code the nanodiamond with a specific biomarkers that help us to direct the nanodiamond to this specific cell. The limitation that we have with our instrument is that we just have one laser in our instrument, so it means that we cannot differentiate between the different populations of cells that coexist in the same sample. So we have to measure in the standard confocal before and try to do some kind of correlation. But this is something that we are in the process to test right now. We are working in a project with PBMCs, and we are trying to differentiate between the different populations of these of those PBMCs. So I can tell you more maybe in
In few
another
months.
presentation.
Great.
Yeah, in few months where we have some results on that. Yeah, but this is something that we also have in mind to check how we can do this kind of differentiation. But currently, it's not possible. Yeah.
Can I ask one more quick question?
Jonathan, please.
Yeah, go ahead. Yeah.
Just a quick one. What do you think the free radical concentrations are in the cells that you're working with? And have you compared those kinds of measures to, you know, some other measurement of the free radical concentration?
Yeah, sure. Usually we use the standard. Usually in the experiments, we measure the amount of reactive oxygen species, for instance, with the standard method, with DCFDA, DHE, or any of the standard methods that are commonly used to detect the free radical, in this case, the Rosa species, because there are not a specific standard method to collect the free radicals itself, but we can measure ROS or nitrogen reactive species as well. So that we do is compare based on these, compare with these standard methodologies. So the amount of free radicals usually, as I mentioned during my presentation, usually they are the amount in are around in the nanomolar range. Usually, I know that the superoxide ion usually has a concentration around the 2 to 10 nanomolar. So that's why also our instrument is we can see that the sensitivity is really high because we can detect this kind of superoxide. But but this is how we do it usually. We measure and compare with the standard method, how we have also the measurement of the whole amount of free radicals that we can find inside the cell.
And so that concentration, I mean, you mentioned doing this control where you measure the response to gadolinium.
Yeah.
And the sensitivity to gadolinium is in that same kind of a range, I guess, or an even more
indeed.
dilute range than nanomolar.
Yeah, when we do the test with the gadolinium, let me check if I don't know if you are
I
still
came in
seeing
a
my
few minutes
presentation.
late, so say that again. I came in just a few minutes late, so maybe you talked about this and I missed it.
No, I didn't talk. I don't know if you are seeing my presentation now.
Yes, I am. Mm-hmm.
Yeah, so this is the gadolinium calibration test that usually we do. So here that we do is test first in a millicube water, and then that I do is prepare different concentrations of gadolinium, and we do it in the range of 1 nanomolar to 10. So as you see here, we have this threshold between 1 to 10, and also 100 or even more if it's necessary. But here in this specific calibration curve, usually I measure in the same range of, in the same nanomolar range of gadolinium. So that's why then we can compare the concentrations in the physiological level in the cells with this standard curve. Because unfortunately there are, yeah, go ahead.
I want to jump in though, but gadolinium is a pretty different spin system than, you know, these individual like free radicals that have one electron, right? So how can you compare the two actually?
Yeah, so this is the standard that we usually use because we want to test the strongest signal that came from a well-known paramagnetic species, because there are no any other a standard that, for instance, we don't have a standards of superoxide ion, for instance. Is not there are not exist any other reagent that we can compare with this, because I think that we are agreed that the best way to do it is using the same standard, because this is the commonly process that we use in the analytical method. So basically use the standard that we want to measure. This is the correct and the let's say the robust way to measure and do a correct calibration of the process. But in this case, it doesn't exist. We don't have any standard to measure the specific ion or the specific radical species that we want to measure. So that's why we use the most related species that we have. Indeed, I agree with you that the spin state of the gadolinium is different. But also this is the sensitivity that we can measure and the reagent that we have available to do these kind of comparisons.
Elkin, why you don't? don't use temple nitroxide radical.
We didn't test that one.
TEMPEOL.
No,
There
I
are
didn't use that
stable.
one before, but could be one that I can use to test. Sure, thank you.
Jonathan, I think you started to ask something. Please continue.
Well, I was going to suggest tempo also. So,
Okay.
Okay.
Then maybe it's.
I mean, I don't really know what to expect of, a high spin gadolinium center versus tempo, but it's probably proportional to the number of unpaired electrons or something like that.
Yeah,
So,
probably, yeah, it's maybe 10 times sensitivity is dropped. It's much more difficult to measure tempo than gadolinium, but it's most relevant, right? Then
yeah, I mean, these things seem very important to me. There's a lot of people relying on these kinds of measurements, T1 and T2 and zero field splitting and so on. And I think doing a robust correlation with a real chemical change that you understand is pretty valuable to the community of people working on this stuff. So this is really nice. Thanks for the nice presentation.
yeah, it's great presentation.
Thank you.
Thank you so much.
Thank
Yeah,
you
thanks.
for the
This is a
discussion.
very
Also,
great overview.
yeah, and thank you
Very
for
good.
your
Okay.
also for your advices. For us, gonna help a lot. Maybe just measure the same this kind of standard. I gonna do it, and I will let you know in the next
Okay,
presentation.
sounds
Sounds
great.
good.
Yeah.
Okay, thank you our audience for very active participations, a lot of questions, and more people will see the recorded presentations. And Elkin, thank you so much again for your very nice presentation, very insightful, and we all wish you luck with your commercialization efforts.
Yeah, thank you so much, and we are open to also to have talks in your labs or in your places if you want to, and also open for scientific discussions as well. Thank you so much.
Okay, great. We will look forward to it. Okay, bye-bye.
Bye bye.